Thank you, everybody, for attending our BioRender 101, an introduction to our software at the University of the West of Scotland. Just for a quick notification, Zoom loves to indicate that you only send messages originally to all of the panelists. So if you do want to chat and say hello to your fellow UWS colleagues, you just do have to click on this blue drop-down arrow here and change it to all panelists and attendees. So that's always a little bit of housekeeping I like to give. It's just me today. I don't have any other BioRender employees monitoring the chat, so I will do my best to keep an eye on the chat and the Q&A for questions. But feel free to put it in the chat, and I'll happily answer it in real time as we're going. Just a quick note, we are recording the session, so I can send out the recording after the fact, so everybody has it in their on their computer, so they can rewatch it at their leisure. Or if for a new individual joining BioRender at UWS, we can just forward them this video, and that will quickly and easily give them the same information that all of you have here today. So let's get started with our BioRender 101 today at UWS. Just a quick note, I've done a quick little screen capture here of what the portal looks like for UWS. Essentially what the portal is it's a unique URL link. So it's located up here at the top, app.biorender.com/portal/UWS. And so this is going to be your home base. Yeah, afternoon. It's morning for me here, but good afternoon to everybody there. And so if you do not have a BioRender account already, signing up through this portal gives you immediate access to BioRender Premium through UWS. If you already do have a BioRender account, but it's not associated with UWS, just below this part here on the screen, sorry, I failed to capture it, there is a tab that says already have a BioRender account, log in. So we just ask that you log in using that. And again, that will integrate your account over to UWS. I'll also be showing this URL again at the end of our meeting, so don't worry about having to copy it down now. You will see it later on in the video. So what we'll cover today is I'll give you a brief introduction about BioRender, and then also we'll switch over to the application itself, and I'll give you a brief demo, and we'll go ahead and create a couple quick figures together. So a little bit of information about myself, just in case anybody was interested. My name is Joseph Zeppa. I am currently a science solution consultant and customer success manager here at BioRender. But essentially what that means is that I'll be your main point of contact at BioRender. So if you have any questions, comments, concerns, I'm always available for you here. And my email is just joe@biorender.com. Simple and easy for you. Feel free again to send me over any questions or comments that you might have. I do have a relatively extensive academic research background. So if any questions or comments you have, actually regard your research, I probably am relatively well versed to understand that. I did my PhD at the University of Western Ontario in London, Ontario, Canada. And I did that in a murine nasopharyngeal infection model of group A streptococcus. And then I did from about 2017 to the beginning of 2020 a postdoctoral associateship at the University of Pittsburgh under Dr. Joanne Flynn, studying tuberculosis in a non-human primate model. So really excited to, use my academic knowledge to help and assist in any way that I can as well. So our story really begins with one of our co-founders, Shiz Aoki, who is the lead scientific illustrator at National Geographic for almost a decade. And so she was able to make these beautiful scientific renderings and share them with the world through National Geographic. But what Shiz realized was that some of the individuals who really needed the ability to do this, such as researchers, scientific educators, and scientific communicators, lacked the tools or the funds to be able to create these beautiful scientific illustrations. Another thing that Shiz and the rest of our co-founders realized was that a lot of scientific concepts could kind of get muddled up just based on the quality of some of the figures that were being presented. So what you're looking at here, I'm sure all of you are aware, is kind of the classic immunological synapse. So an MHC molecule on the surface of an antigen-presenting cell or a cancer cell interacting with a T cell receptor on the surface of a T cell. But hopefully, as you can notice here, Even though these are all essentially the exact same concept trying to be displayed, we have a wide variety of resolution and a wide variety of scientific and artistic abilities coming together here. So we have something that looks like it was maybe made in Microsoft Paint, something that was maybe made in PowerPoint, to a couple other figures that may have actually been created by a medical illustrator or somebody who has some level of artistic and technological savvy. But of course, not everybody has access to, that diverse repertoire of skills. So another thing that was noticed by our co-founders was that, even though there's not only variable, quality of presentation, but all of these events here were trying to represent the same thing. So there was a major lack of standardization in scientific communication and illustration. So wouldn't it be great if there was an easily accessible software that could actually also convey the same information but was accessible to everybody? And all of these things coming together are essentially what created BioRender. So for those of you who don't know, BioRender is an online software application tool that allows you to quickly and easily create your most beautiful scientific illustrations in little to no time and with little to no artistic skill. So all you really need is an internet connection and a web browser, and you're able to log in and create your most beautiful scientific illustrations ever using our icon library of over 30,000 accurate scientific icons. So as you can see here, it's essentially a drag and drop format. So you search for the icon you're looking for, you pull it out, change the color, orient it on the screen as you will, and your figure is complete. Of course, we like to list a couple common use cases here for BioRender, but of course, we also like to highlight that wherever you think you can use BioRender, we highly recommend that you give it a try because I guarantee that it will make your life easier in almost every way possible. So again, some of those common use cases include publication figures, lab meetings, research proposal and grant applications, poster figures, experimental overviews or outlined, and of course, much, much more. So realistically, our goal here at BioRender is to give every scientist the tools and practical knowledge to visually communicate their research. And essentially with that, let's jump into a quick demo of how you could sign in to the software as well as use it to generate some beautiful scientific figures. So I'm just going to exit out here. Hopefully everybody can still see my screen. Can I just get a confirmation in the chat box so I know that you can see biorender.com? Perfect. Thank you so much. So again, as you'll see here, this is just our biorender.com homepage. We ask that you sign in through your portal. So again, app.biorender.com /portal/UWS. But I think no matter what, if you just go over here and sign in, you should be all right. And so I'm just going to do just that. And once I do so, I'll enter my credentials. And I'll come right here to our BioRender homepage. So there's a couple things going on here, so I'll break them down for you. So the lower half of this page is your My Illustrations gallery. Essentially what you're looking at here is all of the illustrations that you will have created throughout your tenure using BioRender. So all of these are different figures that I've created or templates that I've adapted over my usage of BioRender. So one of the main things I like to highlight is that you, of course, have options while clicking on your illustration. Simply clicking on it will allow you to enter back into the illustration and make further edits. You can also right-click or click these three dots, and you can do a couple other things. You can, of course, rename it. You can share it with individuals, make a copy. But the one thing I really wanted to highlight here was your version history. So as you're generating your figures in BioRender, I'll show you exactly where the save function is. But what's really important to note is BioRender actually saves in real time. So if you're ever creating a figure or an illustration and you have your internet cut out or your web browser crashes, you never really have to worry about losing all of that hard done work because it saves in about every minute or two. So let's go to this older illustration that I have here. And as you can see, it saves every minute or two. And you can also go back through and look at older versions of that illustration and make a copy of it if you decide maybe you like an earlier version of the illustration than the one that you currently have. So you've got a number of options here. Not only can you create copies from your version history, but you never have to worry about saving or anything happening to your internet connection and you losing all of your hard-earned work. So another thing I really love to highlight is that, we know collaboration is so key in science. And so we've gone ahead and created what we call our team gallery. So at UWS, you're able to create teams. To do that, you just kind of hover over this team gallery, hit this little cog icon, and mine says BioRender team because that's obviously the team that I'm on. But you yourself at UWS can create your own team. So maybe it's a couple principal investigators that are working together on a collaborative project, or just a lab in and of itself. You can invite members to this team, and then everybody who is on this team can then quickly and easily share illustrations between one another. So let's go back to my antibody development figure. I can hit share. Not only can I just share this with anybody via email, and either they'll be able to open it up in their BioRender account, or if they don't have a BioRender account, they'll just be prompted to create one of our free versions. So even if it's somebody who doesn't have BioRender Premium, they can receive the illustrations that you send them. But back to that team share function, you just go up to the top tab, hit share with team, hit send, go over to this team gallery, and now everybody who's on your team now has access to this illustration that you've just sent to the team gallery. So we've also made it so that that individual can't necessarily mess with your original copy. You'll always have that original copy in your My Illustrations gallery. What this does is this uploads a copy to the team gallery that now anybody who's on your team can actually click on this, download a version of that, and make edits themselves, but they'll never impact that original copy that you have. So that's just our way of making sure that everybody now has access to this illustration and they can make edits to it, but your original copy is unimpacted. And then, of course, if you ever feel like unsharing this illustration, you can just right click or hit these three dots, unshare, and stop sharing. And now it, of course, still exists within your illustration gallery, but within nobody else's. One of the last things I wanted to highlight here from this bottom portion is just the quick and easy ability to, organize things as you see fit. So you can go to these folders, you can hit a plus either there or down here, and you can create one microbiology. And if anything here, oh, maybe this one sums well with microbiology, you can organize things that way. So, we like to give you the ability to customize and organize your illustrations as you see fit. So probably the two main things that set BioRender apart from any other illustration software out there are both its BioRender templates and its BioRender icons. So let's go into the templates first. So essentially what these are these are pre-generated scientific illustrations that you can use for whatever purpose you see fit. So if there's something that you're interested in, so maybe parasitology, so you can just even start to type it, and it'll start to pop up with different aspects of whatever topic you're searching. So maybe you see this malaria transmission cycle and you think that it's awesome and you want to use that in a lecture or in a publication, you can do so. And you can in fact click on it and then use it as a template, and it will essentially become an illustration in your My Illustration gallery. If you want to make edits to it, you can feel free to do that, or you can use it as is. So towards the end of our demonstration, maybe I'll quickly go in and show you how easy it is to edit one of these templates to make it, essentially whatever you want it to be. But I just want to make sure that everybody here is aware you have access to this BioRender template gallery, and you can feel free to use it at your leisure, whether or not you want to make adaptations to one of them or you want to use it outright. They're there for your use. But I think the main reason why everybody's here is, of course, to, take a look at the actual interface itself. So let's go into making our first BioRender illustration. So let's go to blank illustration. We can go up here to the top left, and you can name this. Of course, you can just leave it as untitled if you want, or you can go like my first illustration. Don't worry about naming it. You can always rename it at any point in time, however you see fit. But again, it's always nice to throw something in there right off the bat. So we also have a few options here for the size of your canvas. So, and we've given you a couple common use cases. So maybe you're creating a figure for a journal. We have, a 5x5-inch canvas for that, a figure for a poster, a 10x8, or we find that this 10x7 works really well when you're integrating it into a PowerPoint or Google slide. So I'm going to start with this just because I like the layout of the 10 by 7. But of course, you also have the option to create a custom dynamic image with whatever length by width slide size you want. So I'm just going to hit create illustration here, and I'm going to zoom out a bit just to give you the full effect. So what you're looking at here is our canvas view. So what we like to think of this as is a blank piece of paper on a table. So anything you include within this white space here will be in your final exported image, and anything to the outside here, this gray space, will be your workspace. So you can include anything over here, mock-ups of your icons and your illustrations. Maybe you want to import in a figure to reference when you're creating your illustration. Anything that exists within this gray space will not be in your final illustration. But we always like to let people know that common use cases include, uploading a picture and having it there as a quick and easy reference. So a couple other, tools of the canvas before we hop into creating our first figure. You can zoom in, zoom out. We have these rulers here, that gives you, that 10 by 7 size frame that I mentioned earlier. You can also hover over the ruler and create your own guidelines. So maybe you want half-inch guidelines that you can quickly and easily, add that little bit of buffered white space that really makes an image jump out and easy on the eyes. So you can, of course, click and add those in. And if you start to add a couple and you're not, happy that they're there, you can right click on them and delete them themselves, or you can right click on the ruler line and you can either lock or delete them all at once. So, just a couple other advanced kind of orientation features if you're interested. I'm going to leave the guidelines off for this purpose. We also, if you're used to working in, Photoshop or any other software that has guidelines, you can add those in, make them bigger, make them smaller. And a couple other little notes in the bottom right. We have a bit of a help center down here where throughout today's demonstration, I'll be quickly and easily kind of, manipulating icons. I'll call out those keyboard shortcuts as I'm using them, but just in case you're interested, there is a full summary here of our keyboard shortcuts that I always recommend you get to know because it'll help you, of course, saving time on making your figures. We also have an FAQ help guide that's available for accesses here as well to answer simple questions. We actually have a live chat function whereby we have individuals at the standby waiting for your questions. These are from 9 a.m. to 6 p.m., but this is Eastern time because I am in Toronto, Ontario, Canada. But any question that you do deposit into the chat box is saved and they will answer at their earliest convenience. If they're on chat, this will be within less than 2 minutes. But of course, because of the time difference, it may have to wait till the next day. And of course, the last note there is you can always email me a question as well. I'm always happy to answer anything that you have. So let's get into taking a look at a couple of these icons. So over here, just hit on our icon tab and, maybe I want to start looking at something like a mouse because I use mouse models in my research and I just really want to see what kind of mouse icons we have here. So hopefully you'll notice we have a, wide variety of mirroring icons as well as they're in a number of different orientations. So let's just pull out two different ones here. We've got our kind of anatomical view and we just have a, normal mouse acting as mice do. So hopefully you've realized two things right now. One is that this one comes in a blue box and this one comes in a purple box. And the difference between those two is relatively important. So the blue icons are our just normal icons. They're vectorized images, so you can skew them as big or as small as you want, and they'll never lose that resolution. You can zoom in on them, etc. And again, they keep that nice, crisp figure resolution. Now, these purple bounding boxes also keep that crisp resolution, but hopefully you'll notice as soon as you kind of blow them up, you'll also see this double click to edit group. So let's just zoom in. And if you double click to edit this group, you'll see that the rest of the screen grays out and you can now select actual individual components of this icon. And so that's what the purple bounding box means. It means it's what we call a grouped icon, and each component within this icon is now editable. So, maybe I'm only really interested kind of in the main organs in this animal, and we don't really care about the esophagus or the palate. So I can actually click on those, hit the delete button to get rid of them, and now I'm only highlighting the area that I'm interested in. Another cool feature I love to show here is our circle crop with zoom function. So what that means is you can just type in zoom here, and you kind of get these cool little call out circles. So what they kind of represent is, maybe you want to highlight, a specific portion of this animal here. Maybe we want to highlight these inner organs. So what you can do here is you can kind of see that I'm highlighting a smaller portion and zooming in on it. So what I'm going to do here is I'm on a Macintosh computer, so I'm going to hold the Option key, which allows me to duplicate this animal quickly and easily. And I'm going to now zoom in on the same portion that I kind of have highlighted here with this animal. So as you can see here, this looks obviously kind of funny because we have now a giant mouse on our screen. So this is a perfect call out to our crop function. So if you hit crop, you of course have your classic square crop, which would allow me to grab at any toggle point and kind of make this a square. But we also have a circle crop whereby you can kind of highlight the area, much like highlighting the zoomed in portion here. And if I now click apply crop, we can just orient this a little bit nicer in our circle, and it looks like we're kind of calling in to the inner organs of this mouse. So hopefully that's a bit of a, nice little example of some artistic flair you can kind of give to an icon. But, just as we go, not only do we have really nice icons for, different animal models, maybe another one, C. elegans, I don't even finish typing it, and we have, a nice figure here. Maybe you're working on, you know, immunology, so maybe we can type in T cell, and we even have it down to the cellular level. Same idea. We kind of have some generic natural killer T cells, things like that. Non-human primate. So you can type in, actually, no, let's just type in human for a second, and you can see we actually have some beautiful, human anatomical forms. We have the same kind of inner organ capacity that we do with our rodent. We have different ages, different genders, nondescript genders, anything that you can kind of, think we've probably gone ahead and also thought of and, tried to make this as beneficial to you as possible. Maybe you want to look up malaria. We have a couple of the different life cycle phases here. And then maybe just the last couple ones. Maybe if you work on non-human primates, you can type in monkey, and we've got a nice rhesus macaque. So hopefully you'll see here that we have, a number of different icons from the cellular level, all the way up to the different rodent models, things like that. So, I always recommend that you search any icon that you think you might need, and take a look and see if we have it, and see if we have it in the orientation that you want. Another important feature that we actually have that comes with your premium access at UWS is this request a custom icon button. So if you do end up searching for something that we don't have, but you want us to incorporate it into our icon library, you just go down here to request a custom icon. You fill out the icon name. So again, I'm just going to kind of go in here with gibberish so you can see a couple of the next pages. We ask that you provide some key features, a description, if there's a reference point, and most importantly is if you have any kind of reference image, even just a JPEG pulled from Google, anything that will help our medical illustrators create the best icon possible, we ask that you include that. And then from that point on, within, I've seen them done in as quick as, same day, but we, of course, probably have to realistically say that it takes about 72 to 96 hours just because we've also had some people ask for some pretty complex images. So we do have that appropriate buffer zone, but usually it comes quicker than that. But anyways, with your UWS custom premium membership, you actually each get 3 requested icons per individual per calendar year. So sorry, not per calendar year, per year of your actual premium membership. And so again, if there's an icon that you actually do not find when you're searching through our library, we recommend that you request it and utilize that feature. So I've pulled out kind of a bunch of, nondescript figures here, but let's actually go into creating something that's a little bit more of a complete figure. So I'm just going to, drag my box over all of these icons, hit delete, and get back to a nice blank canvas. So maybe I'll do a quick signaling cascade here. So I'm just going to type in membrane. So we've got these beautiful membrane brushes. So I'm not, I'll drag one out just so I can show you. But realistically, what we were thinking here at BioRender is that, when people are creating things like, monolayers of cells or phospholipid bilayers, either bacterial or cellular or nucleic acid, we really wanted to give people full customization. So anything you see with these little brush icons constitutes our bio brushes. And so these are essentially icons that allow you to give full manipulation of any one of these biological entities that you pull out. So before, if you wanted to create a long chain of DNA, you'd have to go into your icons, type in DNA, find the one you want, pull it out. And then if you wanted to make it longer, you could try and skew it, but that doesn't look great. You could skew it sideways, but of course it would be bigger and that wouldn't look great. So you might have to do Command C, Command V, and copy and paste it a bunch. And of course that was kind of, possible, but that was really time consuming. So what we did instead here was we created our bio brushes. So you'll notice there's a couple different aspects here. We have these blue nodes and we have these white nodes. The blue nodes on the end allow you to grow your DNA or any entity in real time. These blue nodes allow you to have complete custom flexibility in this entity. So you can see, it looks like it's a little bit choppy as I'm manipulating it, but as soon as you let go, it recalculates and gives you that nice, beautiful, smooth flow. And then the last thing are these white nodes, which allow you to add more blue nodes. So if you really need some weird bendy shape, you can, of course, keep adding different layers of bend by clicking on these white nodes and incorporating that in. So another important thing to note as well is that you can double click on these blue nodes to get rid of them. So maybe you created one too many bends and you kind of like it in this more simplistic format. It's kind of quick and easy to do that as well. So I really wanted to highlight these bio brushes because they're, such an amazing feature of BioRender. And in the premium version, you have access to every one of our bio brushes, whereby in the free version, you don't. So again, we like to highlight the features that you now have access to that you, wouldn't have access to if you just use the BioRender free version. Of course, I wanted to mention as well, as I'm going through this, if any questions are being stimulated or anything like that, you can just throw them into the chat box and I can see when a question pops up and I'm happy to answer them kind of in real time. So another little piece of information I wanted to give you there as well. But for the purposes of this demo, I'm just going to use one of our pre-made membranes because I kind of like how simple and easy it is to just pull out and use. I'm going to use my crop function again to just trim off some of this area. So you'll see I'm kind of overlapping a little bit. So some of this image is in the white space, some of this image is outside of it. I like to give myself a little bit of overlap, but I know that, of course, anything outside of this black line won't be incorporated. So I'm actually not too concerned. Another awesome feature of BioRender that I love to mention is our lock object. So maybe I'm going to throw in a receptor here, just like this. And this is the receptor that I want to include on my surface. Maybe in the future, as I'm kind of starting to manipulate things, I go to grab my receptor, but I accidentally grab my background and I skew it to the side. And although it's just a simple Command Z to undo and get back to the previous step, doing that all the time is a bit of a pain. So as soon as you have something on the canvas and you know that it's in the orientation that you want, I always recommend that you lock it. And that way, you no longer have the ability to move it from its spot. Of course, you can still select this item and you'll see that it's locked and you can unlock it, but that's also just kind of a nice feature that I do like to highlight to people. So, let's kind of continue along with this. We're going to create a bit of a signaling cascade here. We've got our receptor. It's one of our grouped icons, so I can actually, ungroup it and I can see that we have this, beautiful molecule. So I'm going to hold the Option key to copy this. Maybe I want to indicate a bit of a flow here where I have this molecule coming in to bind this receptor. So there's no better way to do that than by using an arrow. So I'm going to go Insert Line. And then here, we've kind of got these 5 different arrow types that all are very unique. But what's really nice is any one of these line types can actually be turned into any of these other top 5 line types. So, maybe I want to use this arrow with a faded tail to denote movement. You'll see it's here just as simple as clicking and dragging, from the beginning point to the end point. And we've got this beautiful arrow here. But if I figured out that I wanted this to actually not have a line fade or change the color, as soon as you click on this arrow, you'll notice this beautiful toolbar that pops up. And you can adapt things like the line color, the width of the line, whether it's dashed or not. You can toggle on and off this line fade. You can also switch orientation very easily, which is one of the features I love about our arrows. And of course, as I mentioned, if you decide maybe afterwards, oh, actually, we want this to be an inhibitor line, not an arrow, instead of deleting this and inserting a new line, you can just go to the head here and change this to an inhibitor line. And that's how simple it is. Much like our bio brushes as well, these blue nodes do the exact same thing as they do with our brushes. So you can, increase or decrease the flex of the arrow. And of course, if you wanted this to be, a very wavy arrow, you can do that as well. Of course, that's not something I'm interested in today, but our arrows are highly, highly, highly manipulatable to give you exactly the arrow shape and size and color that you want. So again, our line colors are really cool. We've got a bunch of these beautiful presets that you can actually see. It changes them in real time. So you can kind of see what it looks like before you actually make your selection. And if you know a specific color that you want, the RGB or the hex code, you can freely do that. Or you can also use our color picker to steal a color. So maybe I want to steal the same color, as my molecule here. I can quickly and easily kind of do that. So, the color picker really gives you the ability and the, function to change the color to whatever you want. I'm going to go back to a black line, but, that's another really cool feature that I love to highlight. So let's kind of keep things going here. And let's say we want, this molecule binding, causes this receptor to, oops, I just realized that I didn't regroup these things together. So let's group them back together here. There we go. And, now we have our receptor being endocytosed into this little vesicle here. So maybe I want another vesicle to show that's now going to be actually brought into the cell. So let's bring in this little guy. Colors are a little bit different here. So I'm just going to go up here and quickly change my icon color. And I'm going to now bring this again into the vesicle. So you'll notice now I've brought in a few different layers of items, and now my receptor is behind my vesicle here, which doesn't do anybody any good. So another important thing to note is that you can, of course, right-click on any item, and it brings up a number of different objects. You can flip, you can lock the object this way, and you can also enter the group or ungroup it. So what's really important here is that I want to go to my arrange function, and I want to send this to front. And that way now the receptor is exactly where I kind of want it to be in my flow of objects. Another thing that's really cool, so, clicking on this object, you saw that we can ungroup and regroup this object together, but maybe I realize that, I want to consistently use all of these objects together kind of as my, future idea of having a molecule bind a receptor. So I can, group them together, etc. But one really cool feature I also want to highlight is my favorites feature. So highlighting, I was just holding shift there to select all of these items, the arrow, my molecule and receptor, and then the molecule itself. Hitting this favorite button, hopefully you'll notice along the left-hand side here, allows you to look into your favorites menu. So what this is this is essentially, think of it as a curated playlist for you, but a playlist of your favorite icons. So instead of having to instead of having to search new icons each and every time that you want, if ever in the future you realize that, you're starting from your membrane again and you really want that icon instead of having to draw out a new arrow, search for a new molecule. Although that is relatively quick, you can save even more valuable time by favoriting icons you use a lot and finding them there. And then now you can just easily, quickly, drag things out. And now it's exactly where you wanted it to be. Another important thing, much like you can team share your final illustrations, you can also team share these favorited icons. So, maybe I've created a bit of a complex, scenario here that I want to share with my team so they can use this in the future. I can go up here to the top left, go share with team, make sure this box is checked on the team that you're interested in sharing with, go update, go back to team. Now my whole team has access to this combination of icons. And of course, you can also unshare with your team if you want to do that as well. So hopefully that gives you a good idea of your favorites little tab here and the fact that you can share that with your team. So let's go ahead and kind of finish this image off. So let's go back to icons. I kind of want to include a nucleus here. We've got a nucleus brush, but for simplicity, I'm just going to pull out kind of one of these preformed nuclei. I'm also going to just pull out kind of some preformed DNA here. Let's do this one. Looks pretty good. There we go. And then maybe we'll kind of start to follow this through. So we're going to say, the molecule binding causes some, endocytosis here. Let's go like that and have things snap out a little bit. Then it also causes this event, which is our formation to our intracellular vesicle. And then this goes down to, let's do something a little bit more janky now. There we go. So let's do this. And then this kind of goes through here to our actual, signaling cascade. And maybe it causes a gene to be transcribed. And then with that gene being transcribed, maybe we now have a protein being produced. So we can kind of go insert line. You can just have this going kind of up from the nucleus. And then we can kind of also just go, I'm just going to go nondescript protein here. That green's a little bit much for me. So another cool thing is when you're searching icons, if you see one in a color that you're not a big fan of, you can always change the color up here, but also clicking on the icon itself gives you an idea of, the different types of molecules you can actually create. So, maybe we want this one we seem to have a bit of a blue theme going on here. So let's kind of keep that alive and well. And, we'll see we're producing now this kind of relatively large protein. Another thing that I realized here is, we've got a nice arrow flow going on here. So actually, I don't like this arrow type. So maybe I'll switch it to a bit of a wavy one. And we can kind of get rid of a couple things there. There we go. It's a little bit better. But what we realized is we haven't really, numbered these steps or anything. So this is a perfect opportunity for me to bring up our text shapes. So text shapes are something that we, realize that a lot of other softwares have and that we need to also incorporate, but it also saves you a lot of time. So, you can create these kind of classic protein pill shapes if we were denoting some kind of, maybe signaling phosphorylation cascade, but they're also fantastic for numbering your steps. So, maybe here we have step 1, which is binding. So I can also insert text and I can go binding. So after I add text here, hopefully you'll note that you can kind of quickly and easily put your text down. And you can, of course, change let me just move over a little bit here. So you can see that we can change the type of text we have. We can change the size. We can also bold if we wanted to. We can change the color, much like we could the color of our lines. And of course, you can justify the text any way you want. If you actually double click into it, you'll notice that we also have the ability to insert specific symbols, all of your Greek and alphanumeric characters that you're used to. And you can also add superscript and subscript. So hopefully this is, kind of a good idea of, an easy way to denote signaling of, or events. So we have binding. We have maybe number 2 is going to be endocytosis. And then maybe we can go number 3 is something like translocation. Sorry, I'm just going to check questions here. My apologies. I did miss something. I figured I did. Oh, love that feature. I'm so glad you love it. And then maybe number 4 is something we'll do, protein production. And you'll see it kind of knew that I was at the edge of my screen there. And so we can go like that and, looks a little bit nicer. So, what I really love here is we kind of have this flow that was quick and easy to throw together. A couple other last minute features that I will also show you are our PDB integration. So maybe we'll make this a bit smaller, but maybe we'll use that zoom callout again. And, we'll say that we're trying to let people know kind of what this protein is here. So you kind of put the first circle there. Maybe we'll orient this up a little bit so that we're not crashing our membrane as much. And another thing you'll note too is, maybe I need a little bit more white space up top. So as I mentioned before, you can adjust the canvas size pretty much at any point in time. Just click on the kind of the background, the canvas size, go up here to canvas size, and then much like our crop function, you can kind of quickly and easily just adjust the size of your slide a little bit. Therefore, you've got now a little bit more room to work with and kind of manipulate. So our PDB integration, again, I'm sure that everyone here is aware, but the Protein Data Bank, or PDB, is a repository of three-dimensional crystal structures that have been published in the scientific literature. And so any crystal structure that's in this data bank, you can actually import and bring in to your BioRender illustration. So you just go up here to create protein. If you know the 4 alphanumeric character code for your PDB molecule, then enter it in there. I don't have one off the top of my head, but maybe we will, use this 5IRE. And so this is our cryo-EM of the Zika virus. So I know this isn't very specific because we have a protein being created and this is a Zika virus, but hopefully what you'll see here is, you've got this beautiful 3D orientation of the Zika virus. The controls in the PDB integration here are right here in the bottom right, so you can kind of always get a good idea of, how to zoom in and zoom out. This seems to be a relatively large PDB icon, so it's a little bit slow, especially when I'm streaming on Zoom here. But, it'll probably be a much smoother interface if you use it just on your computer alone. So, of course, you can switch between cartoon, licorice, and van der Waals forces, of course, because this is such a busy molecule, maybe cartoon and licorice aren't really a good idea. But you can, of course, also change the colors, based on molecule, sequence, chain. Chain looks pretty cool, so let's keep it that way. You can adjust also the border style to give it maybe a little bit more three-dimensional depth. And you can switch between flat or kind of toon shading. So I always like the toon shading here. So, maybe we want to keep our Zika virus, little protein ball here, save the icon. And as soon as you save the icon, it just automatically pops onto your screen here, and you can kind of orient it as well into our zoom callout. So this is a pretty cool feature that I always love to highlight, and it kind of gives you a really nice idea of, how to kind of use our zoom feature, not only to kind of zoom in as we did with our mouse example before, but also you can kind of use a general nondescript protein in your actual image, but you can also highlight that you are aware, of course, of the complexities of that molecule. Yes, increasing canvas size is always a super useful tool to have. So with this, I'm going to actually, this will be the image that I export, but I do want to quickly go in and show you how to edit one of our templates simply and easily. So what you'll also hopefully notice is that I didn't, again, save once throughout that entire experience, and you'll see that, my image is right here in its completed form. So there's kind of hopefully a real-time example of our, version history saving in real time. Can you import images, say SEM? So exactly. So what I was going to highlight here with my BioRender template. So, let me just type in mass spec. Maybe I want to adapt a, mass spec protocol here. There's one that I love to use, which is this one right here. And that's because it highlights a couple really cool features. So I think the question was, can we import different images, PDFs, SEM images, etc. So that's the next thing I was going to actually bring up here was, so say, we have our neuronal tissue here, but we want to include maybe a histological slide. We do have an upload feature right here along the left-hand side. So you can import images, icons from different companies or UWS. You can import in graphs from GraphPad Prism, and you can import in, any sort of items that are JPEGs, TIFFs, PNGs, and SVGs. So those are the 4 file types you're able to import into BioRender. So anything else you will have to actually convert to one of those file types, but you can import those 4 very common image and vector file types. So again, I kind of already have this in my library saved, of course, of which you can also share with your team, much like your favorites. But you can also pull those in, and now you can, show that you've got, maybe this is our histology slide of this neuronal tissue. So another thing that I like to highlight towards the end of our demo is our preview in grayscale. So, off the bat, this might seem like a weird thing to do. Why would I ever kind of want to look in grayscale? But it does 2 things. Number one, it's a great idea always to take a look at how potentially your visually impaired audience might observe your photo. You might lose a couple very key points that you're trying to make, and you want, of course, this information to be accessible to everybody. But the second thing, equally as important, is contrast. So say here in this sample, I wanted to say, okay, well, this is just kind of a, a petri dish. Maybe I want to include some cells. So I type here and I type in cells. And, we've got this nice cluster of spheroidal cells that I pull out, and I'm going to shrink them a bit so that they fit on my plate. And then I'm also, very rarely do I recommend that you skew things, sideways or horizontally or vertically, because it kind of, doesn't make it look great. The one caveat to that is, whenever you have a plate that's kind of tipped, at a 45-degree angle back this way, you can flatten your images onto it like this, and it kind of looks like the cells are now on the plate. So one of the very rare circumstances where this actually looks, okay. Maybe I look at these cells and I say, oh, purple's my favorite color. Let me throw purple down on this. And, purple, red, very different colors, and I've kind of got purple and red here. So it works out well. This is where preview and grayscale really comes into play because, without looking at this, I would think that this actually has very good contrast. But in switching back now to my preview and grayscale, I note that the cells are almost indistinguishable from the background of this, circular culture flask. But if I go back to that original orange color, I notice how much more these cells pop on top of this darker background. You can actually scan through almost every color and see which ones have better and worse contrast. And of course, ones that you think will be bad are usually bad, like any dark color on dark color, kind of black on that darker red. But you'll also note that there's definitely better and worse colors. So let's go back to that orange now, because you can see the contrast looks really beautiful. And lo and behold, it actually, now that I've double checked it in grayscale, I actually do see that this orange color does look better. So hopefully that's a couple quick examples of how, maybe I now want to, with my data analysis, upload again my own GraphPad Prism file here. And so again, I exported this at relatively high resolution, so you can blow this up. You can steal the colors from it if you want to change your text here. Maybe you really like this dark green. Oops, I accidentally stole the white. That doesn't work. But anyways, hopefully you get that same idea, There's that dark green. There you go. But anyways, so hopefully you can see now that you can kind of import your data graphical images from GraphPad Prism. and your histology slides, and some importance of preview and grayscale. Let's go back to the gallery and let me export this original image here and show you how quickly and easy it is to integrate into a Google slide presentation. So a couple things along the top. Preview is always important. You can take a look at what your image will look like kind of in a more almost full screen view here. You can kind of see the flow, make sure everything looks and fits exactly how you want it to. You can then, of course, automatically share with people if you know that you're not going to be exporting this into a PowerPoint. You can immediately send it to somebody via email or share it with your team, going back to that team gallery function. But let's say that we are going to export it and use it in a presentation. So again, we can see here finally, here's our preview. You can actually toggle on that preview and grayscale from this image as well. So it gives you another final opportunity to check your contrast. You can adjust the width and the height in the actual view as well. And of course, your export, you've got 3 different types: PNG, JPEG, and PDF. We recommend PNG, especially if your image, is relatively beautiful, but a little bit, kind of basic. It just has your nice core principles involved in it. And that's because we have this beautiful transparent background, which essentially takes all of this white space in behind and it becomes transparent. So I'll include it in here just so I can show you what it looks like once we import this into our presentation. You also have a few different export resolutions, and a couple of different recommendations for where you would use those resolutions. So we've got 72 DPI if you're using it on a website, 150 DPI if you're using it in a presentation, which is what I'm doing, and we have 300 DPI, and usually that's what's required for journal images as well. So on the free version, you would actually only have access to this lower resolution export. So now with Premium at UWS, you actually have access to all 3 of our export resolutions. So once you're done, I'm going to hit export, and a nice little pop-up is going to come here for me. And essentially what this is indicating to you is that we ask that you do cite that you've created your image in BioRender. So if you are, especially if you're publishing this, we ask that you include this in either your figure legend, if you're only using it in one figure in that publication or one panel in one figure. But if you're using BioRender to create multiple illustrations for a publication, you don't have to worry about putting it every single one of your figure legends. You can just include it in the acknowledgments at the end. Figure created with biorender.com. We've made it even so easy that you just hit copy, and this is now copied to your clipboard, and you can paste it in whatever appropriate location that is. So now that our figure is done and it's exported, so I'm using Google Chrome for this whole presentation. It works in, BioRender works in almost any Internet Explorer or any other web browser. But we do prefer you use Google Chrome just because that's where we do all of our beta testing. So it just, we guarantee, as much as we can that as many bugs are worked out in that platform. So, what's also nice about it is that whenever you export an image, it kind of just pops into your, lower hand quadrant here. I can go back here to my BioRender at UWS. I can go down to this lab presentation slide, and I can quickly and easily drag this into my presentation. And here's that transparent background I was talking about. You can see that, that white space that used to be white space is now transparent. So I think I see one more question here. Yeah, Chrome is the best. Yeah, we definitely use it for all of our, beta testing. So we definitely, recommend it so that you get the smoothest experience on BioRender as possible. So with that, we've got about 4 minutes left. So I wanted to say a big thank you for joining us. Special thank you to Professor Henriquez for helping us organize today's session. It would not have been possible without her. I also wanted to include, there's that portal that I mentioned at the beginning, app.biorender.com/portal/UWS. This is where you can sign up through the UWS for your free BioRender Premium account, or if you already have an account that you want to integrate over, you can do that here as well. Of course, if you have any questions, comments, or concerns, you can email us at support@biorender.com or directly to me, joe@biorender.com. And let me just do one last quick check for these questions. Thank you for the demo. The software is wonderful. Just to be clear, we use images for published graphical abstracts or in our outline teaching without convert. Yes, exactly. So to answer your number one question, because you have BioRender Premium through UWS, there's no copyright issues whatsoever. You have the ability to use your BioRender created figures anywhere you want. And also on that note, they're completely your intellectual property. We have no, there's no back print somewhere that says we own your illustrations or own your information. It's all yours. We're just the platform that you're using to present that information. Can we export multiple illustrations at once? So, it's not quite ready yet, but we're actually creating a BioRender, what we're calling it as a BioRender slide deck, which will essentially be that canvas view that I showed you right here, but along the side, you'll actually have multiple illustration slides in one, and you can export more than one. The feature isn't quite ready yet, but we're very close to launching it. So definitely keep your eye out for that feature. But as of right now, you do have to export your illustrations one at a time. So I do apologize for that. Hopefully that answered your question about kind of copyright issues or anything like that, and uh, presentation publication. Are there any last minute questions we have here in the last 2 minutes or so? It doesn't really look like it. So with that, I really like to say thank you so much for joining us. I will be providing um, a recording of this webinar, so you have it for your future use if ever you want to go back to it, or for distribution purposes to other people in your department who kind of join on to BioRender and want the same 101 experience that you had. And again, with that, thank you very much for joining, and have a fantastic rest of your day and weekend as well.